And our next speaker is Dr. Jack Gilbert, who is recently moved to the University of California, San Diego. So, Jack, all yours.
Thank you. Go to my first talk title. So talk two, well, I'm talk two, which is good. So I'm going to follow up that. I'm not an epidemiologist. I'm not even a clinician. I actually started out my career as an ecologist, working with butterflies, and somehow I got into bacteria. But over the last, say, 10 years, we've been exploring the microbiology of the built environment. Started out as someone gave us $100,000 to do a small study and then somehow spiraled out of control. I'm now at UCSD. I'm in the Department of Pediatrics and Oceanography, which is kind of a weird combination, but it works out for me. So my main interest in this is transmission, the point at which the human themselves become an element, a source of microbes to the environment, and the opportunity for that environment to feed back upon the human. And routes of transmission have been well established for well over 100 years. In fact, the primary literature from this starts in around 1880, with people saying, well, I can colonize human-associated bacteria from buildings. And it's reasonably well affected. In fact, they even talk about it in the Bible. There's several passages in the Bible which deal with human-associated colonization events, and they talk about cleaning it with horse's blood to get rid of dangerous fluxes. So, we've been dealing with the microbiology of the built environment for a long time. But these individual components of either inhalation or fomite-based skin colonization are our two primary indicators for exploring how microbes colonize these environmental processes. And respiratory, we think, is incredibly important. We work in asthma and trying to understand that. I'm not going to go into our asthma work because I don't think it's particularly relevant here. Hopefully, people who have asthma won't necessarily immediately be going into space, but there is always the opportunity to explore strategies to optimize airborne transmission events to reduce the likelihood of colonization via the respiratory system, but also maybe to improve immune stimulation via respiratory-based activation. But my main interest in this space is the transmission routes through the skin. Obviously, Julie's done a hell of a lot of work on the skin, and we've done a reasonable amount of work trying to understand how the skin transmits to the environment and vice versa. It doesn't just stop there, though. Microbes are the best chemists in the universe, we think, and that component also has to be dealt with. We've known for very many years that microbially mediated metabolites, either aerosolized or via skin contact, can play a role in human health. And those are things that we really have to consider. And obviously, this is something that we've been dealing with, and a lot of other people, Rachel, for example, been dealing with in terms of trying to understand the individual components that come out of chemical factors associated with microbiology in this space. Sufficient evidence to understand the nasopharyngeal inflammation, wheeze, cough, bronchitis, eczema, and associated microbial metabolites. We look at skin disorders in space flight. We were hearing about that yesterday. That may be an individual component that could be of concern. Well, microbes, full stop, may not be playing a very significant role, but the chemical products that they produce may be, and that has to be dealt with. In fact, interestingly, when you get down to trying to understand that component, getting chemical modification and chemical monitoring is actually reasonably well established. We can put in place chemical sensors that allow us to do early warning to determine critical load of a particular metabolite in an environment reasonably effectively as an early warning system of buildup. So if we're doing long-term space missions, that's something where we could say, well, we need to target this metabolite, we need a chemical sensor to deal with it. We have done some, working with Jonathan Eisen, done some work on the microbiology of the ISS. Very, very limited work, but a nice first pass. Also, a very good indication of how bloody difficult it is getting samples back and up there. Very small, tight space, having to get like little tiny swabs up into space. Obviously, it looks very similar to what you'd expect, as we heard yesterday, what you'd expect of any kind of human-based occupied environment. And in fact, if you compare it to home environments on Earth, you see very, very similar microbial community similarity. especially when you compare it to non-human-occupied environments. So yeah, I mean, the ISS is a human-occupied environment. It's got a lot of human bacteria in it. And that's reasonably well established, and we can deal with that. But that's the important component, right? It's that flux. Human bacteria get into the environment. Human bacteria in the environment can get back onto the human. My interest is in what happens to those human bacteria when they leave the human, end up in this hostile ecosystem called the built environment, and then recolonize the person. This is preliminary work that we've done with Venkat. We just published earlier this year, looking at the potential of fungi, and we have some work on bacteria coming in to colonize and attack nematodes, in this case, as an indicator of immune activation and fatal colonization. And in this case, space-flown organisms, F3, F4, and 293, seem to show a significant increase in what's the word? Pathogenicity. There we are. I need more tea. In terms of trying to attack the individual organisms. So when we put a fungi in space, take a step back. If a bacteria or a fungus leaves a human being on Earth, we know that sometimes they can become more pathogenic in the environment. In space, that pathogenicity potential may be accelerated. And those are things that we need to take into consideration. So the building is obviously key to what we want to understand, and each one of us are releasing 38 million bacterial particles into the immediate vicinity every hour, around 7 million genomic equivalents of fungi. This is work from, who did that? It was Jordan? Yeah, there we are. But that particular component is an average, just sitting still in a space, right? That was reasonably well established. We want to be able to explore what that impact looks like. If every single person is emitting their own microbial aura cloud that we heard about yesterday, the pig pen routine, then we need to be able to determine that particular trajectory and how it's going to influence the environment, what those organisms are evolutionarily selected for in that environment, and then how that will feed back upon human health. We've done a lot of this. We're trying to determine how that impacts how we design buildings. Potentially, in this case, we want to see how that impacts the design of spacecraft. My primary interest is, yes, those things are going to leave people, they're going to land there, but what happens to them when they land on the surface? So in this case, we took multiple different types of surface material and placed those surface materials into environments where they could be colonized, an environment such as a family home with pets, a home with a single man living by himself, a space that had no human occupancy, and then worked out how the organisms that colonized those materials reacted when the material became wetted. We've known, again, for hundreds of years that you wet the indoor environment, it gets damp, it gets in a high level of moisture content, it will have an impact upon microbial growth. But what we weren't able to necessarily determine is the biochemical nature of the interactions that led to that growth. So in this case, wetting causes growth, we know that. The abundance of microbes correlates very well with the abundance of virus-like particles. Phage and viruses colonize the environment. Phage increases as bacteria increase. Viruses can increase as fungi increase. And we see a significant differentiation in the degree of particle counts associated with different types of materials. Here we have mold-free gypsum, which is treated with a chemical which prevents the buildup of fungi. And we have MDF, straight gypsum, and density board. We know that bacterial and fungal diversity tend to decrease, so 16S and ITS as bacterial and fungal markers tend to decrease over periods of exposure during wetting. Obviously, when you wet it, something has to grow, something is selected for. That causes dominance in that environment and a reduction in observed diversity. Interestingly, though, the chemical profile of those environments shows very little differentiation and change. We do have a huge array of metabolites, chemicals on a surface material anyway, irrespective of it being wetted or dried. The microbial contribution to that is actually quite small, but is detectable. Bacterial and fungal diversity is very well correlated. Pseudomonas and bacillus growth is anti-correlated. When a Pseudomonas grows, bacillus tend to be inhibited and vice versa. Eurotium grows well without water. We actually see an increase over time with longitudinal observation in the density of Eurotium, whereas Penicillium does better with water. Different environmental traits will select for different types of organisms, and especially when we're dealing with, say, a Candida outbreak, we want to be able to determine what is causing the survival of that organism in the environment and what could promote or not promote its growth. These are just three different components: the bacteria, the fungi, and then the metabolites on the bottom, and the different types of environmental traits that associate with them. So, as you can see, a wetted in blue community sample has a different bacterial community, a different fungal community, and to a certain extent a different metabolite profile to a non-wetted environment in yellow. The location, the environment in which the microbial community was colonized from plays a significant role in terms of what actually colonizes the environment. And then the type of material which the organisms are growing on also plays a role, especially in the bacteria and in the metabolites, but not necessarily in the fungi. We see the same kind of fungi irrespective of the type of material, unless you're talking about mold-free gypsum, which obviously wipes out the fungi. We can actually look at the particular associations between particular metabolites and the growth of bacteria and fungi in this environment. Antibacterial alkaloids like nigragillin and fumigaclavin C correlate very well with Ascomycota abundance, but negatively correlate obviously with the abundance of Bacillus and Pseudomonas. Suggesting in this case that some of the fungi are producing antibacterial elements which can actually eradicate the competition they might get from bacteria in the environment. If you have an extremely low nutrient environment, microbes will be trying to clear the way of the biomass that's associated with that space in order to allow for growth. Interestingly, nigranillin was greatest on the medium density fiberboard, and no bacterial growth was detected on medium density fiberboard because the fungi were just eradicating the ability of those organisms to grow. We can do autocorrelative steps, and this is very ugly, but it's informative. Azoxystrobin was positively correlated with Bacillus, but negatively correlated with Pseudomonas. So the Bacillus was actually producing its own antibacterial against Pseudomonas to eradicate the growth of that organism. Once you understand how the engine works, you could tweak it. So if we want to be able to get in there and say, well, we don't want those Pseudomonas around, then maybe we need an organism in there that could eradicate the growth of the Pseudomonas, for example. In this case, we're actually using that strategy right now. We've identified non-lethal strains of Bacillus that can produce azoxystrobin, and we are applying them into controlled hospital settings at the Rady Children's Hospital in San Diego to eradicate the growth of certain types of Pseudomonas that they've seen as being potentially threatening to NICU scenarios. So that's engineering using the microbial community to control that environment. And then scopolectin was positively correlated to Pseudomonas, negatively correlated Bacillus. So, in that case, the Pseudomonas is trying to find a way to eradicate its competition. We applied basic components of this strategy to hospital environments. This is the Center for Care and Discovery, which is a brand new hospital building built in around 2014 in Chicago. We were very lucky to be able to get into that building before it became functionally operational. We collected 365 consecutive days of monitoring in that space, 2 months pre-opening, so 2 months before it became a functioning hospital, and then 10 months post-opening, collecting patient skin and hospital staff after the opening, and then prior to and continuing after opening the patient room, air and water and nursing station sites, providing us with an ability just to do observation over time. You'll see a trend in virtually all of our work. Longitudinal time series analysis, in my opinion, is one of the fundamental ways that we can detect, for example, growth. You're talking about growth. How do we determine if something's viable? Well, if it's changing over time, that's either an indicator that's being eradicated or an indicator of growth. And we can generally disassociate that with a longitudinal sample. In this space, we can see extremely low biomass prior to occupation, so pre-opening here in blue and post-opening in red, and we can see a very significant differentiation. As soon as the hospital becomes a functional environment filled with human bodies, each emitting 36 million microbial particles per hour, we see a significant shift in the abundance of organisms that are most associated with skin, like the Corynnean staph. Versus prior to when we see just a lot of Acinetobacter and organisms which are generally associated with incredibly low biomass and cleaned environments. So that's a significant ecological shift in the paradigm. One of the things that I find most interesting is that we can track genotypes, so metagenomic genotype reconstruction. We can track genotypes of individual types or subspecies of particular organisms, particular staph, particular Neurococcus, for example, and see that those genotypes appear to be showing an increased acquisition, maybe not acquisition or selection, for antibiotic resistance genes over time. So over 60 days of hospital exposure, we see a significant increase in the abundance of ARGs associated with these genotypes over the environment. So at the beginning, you see those genotypes, but without ARGs. After 60 odd days, you see an increase in ARGs in those genotypes on average. And we also see a greater non-synonymous to synonymous mutation ratio in the coding regions of those genomes, which is highly suggestive of a selective environment. If you had non-synonymous mutations, it's drift, right? If you've got synonymous mutations, then you're seeing non-synonymous mutations, then you're seeing a significant selection pressure that's driving the selection of particular functional ecotypes. And that's what we see in this environment. These organisms are under selective pressure. The relative humidity positively correlated with microbial colonization and transmission. Staph were more microbiologically similar to each other during a high humidity summer environment, for example, than they were during the low humidity winter environment. Airborne humidity, airborne water acts as an increased likelihood of bacterial transmission and colonization between individuals, but also with the patients themselves. And we can track that using Bayesian correlation and codependency analysis. So here we have a staph person, staph nose, staph hand, their cell pager, their shoes, their shirt hem, their pager, gloves, for example. And the staph were always a significantly greater source of bacteria to the patient than vice versa. The patient was rarely a source to the staph. Why? Well, the staph is constantly putting on and swapping out gloves as they move between rooms. So when they're interacting with the patient, there is airborne fomite-based transmission from the staph member to the patient, and probably vice versa. But then they take their gloves off, they wash their hands, they go on to the next patient and eradicate that transmissible element, right? So they're never really taking on board the patient's microbes and then moving them around. In an ideal scenario, we talk about like hand washing. Sometimes it ain't that ideal, but when you're observing them, suddenly everybody washes their hands on a regular basis. And we've done this in homes as well. So this is, don't tell the IRB, this is my home. 4 noses, 4 feet, and 4 hands. We can track the transmission of bacteria over time between the individual components. And I'll hold up my hands. We're tracking the transmission, in this case, of 16S elements. We've done it with genotypes in this study, but we are only using about 100 different samples. And so you could look at genotypes being moved between individuals, but it was very limited. With 16S, because we had over 600, 700 samples in this study, we were actually able to track particular 16S sequences over space and time. If you add dogs into that environment, you get a significant increase in the complexity of the dynamics in that space. Couples with a dog will actually share more bacteria than couples without a dog. This is work from Rob Knight's group. Couples with a baby will share less bacterial similarity than couples with a dog, suggesting that dogs actually act as a better conduit of microbial transmission. So I think dogs are potentially important. And dogs in space might be a better way of helping the astronauts to share positive microbial similarity. That's actually my dog, Captain Bo Diggly, on the new beach in San Diego. He found a dog beach. He absolutely loves it. But I do like that idea, right? Dogs were the first animals in space, or was it monkeys? I can't remember. It was a dog. It was Laika, wasn't it? Yeah? So let's embrace that reality. If we're going to colonize something, let's bring dogs along. Interestingly, even without a dog, if you physically interact with an individual, if I was to hug Iran on a regular basis, we would increase our microbial similarity, right? We'll share more bacteria. In this case, we have a couple, person 1 and person 2 in red and blue, compared to person 3, who's the lodger living in their home. And the couple always have a significant increase in microbial similarity compared to the lodger. And that relationship we know is important. Interestingly, the only environment on their bodies which is always differentiated is their nose. I don't know if you ever tried, but it's really hard to lock noses. I've tried. Maybe that's just me. You can actually track what people do as well. So using a source tracker algorithm, we looked at the likelihood that people, these are 3 people, person 1 is a 25-year-old man in red, and then person 2 in orange is his dad, and then person 3 in yellow is his mum, and he's got 3 dogs. And he's living at home. If you look at the kitchen counter, the x-axis is time, the y-axis is the percentage of bacteria found on that kitchen counter that originated from one of the occupants. Obviously a heavily gender-biased home, because in this case 100% sometimes of the bacteria found on that surface originated from the mother, until she goes away for a few days, and the father's microbiome explodes onto the kitchen counter as he desperately fends for himself trying to make tea. I'm not saying who this is. You can see the floors are covered in dog bacteria, and some of the dog bacteria get onto surfaces like the doorknobs and light switches because you are acting as a fomite transmission agent between the dog and those surfaces via your hands. So we can track these activities. We've started to explore the potential to do that, working with Venkat on the microbial and analysis of the ISS, the potential to do that with individual astronauts on the ISS. Can we track which spaces they most interact with? And would that help us to understand better the likelihood of transmission agents within individuals, within different components? And so I was most interested in the differentiation between the Russian astronauts or cosmonauts, and the other international astronauts, and the differential use of space. With a more Russian section and a more international section. I'm very interested to see what if you could predict that and what relationship that has in the microbial context. We just finished a study, a very simple study, but working with air cadets. Cadets, much like astronauts, go through a very rigorous training process, but then they are co-associated and living together from a situation of never having met each other. And then they spend a lot of time together, and they eat the same food, and they live in the same quarters, and they do the same kind of activities. It's very stringent. And Air Force cadets got more similar to each other over time as they lived together from the point of meeting each other, right? But interestingly, this is one dorm room, and the cadets living in that room. And we've, again, used source tracking here. And you can actually see desk 1 was on one side of the dorm room, and desk 2 is on the other side of the dorm room for the 2 people sharing that space. And skin of the first cadet was more similar to desk 1, and the skin of the second cadet was more similar to desk 2. There was bleed over, but we could determine which cadet was using which desk in the room based upon the microbes they left behind. I just think that's fun. And just to finish up, we explore ways of doing this in homes, how to manage the microbial diversity, how to engineer a more healthy home environment with the microbes that are in there, but such as reducing surface moisture for unnecessary growth, including household pets. The dogs are important, maybe not the cats. We don't need the cats. We've done a lot of work on controlling exposure to farming environments. The animal-associated bacteria in a farming environment appear to be very beneficial for health, especially in young children. Obviously not potentially relevant for space travel now, but if we were talking about colonizing an environment and bringing children along on a colonization or a, what do you call it when you take a planet and you make it more human, more terraforming, yeah. If we're going to terraform, we want to bring along some cows and dogs. Did I mention the dogs? The dogs will be important. And then intentional increase in indoor microbial diversity. We heard the astronaut lady yesterday talking about, she could smell the plants and how nice that was in such a sterile environment, right? Well, this is a natural human component. I spent 2 years living in Antarctica, and the The best memory from that trip is when I was coming back across the ocean on the icebreaker. I'd not seen a plant properly in 2 years, and I could smell the eucalyptus as we were coming back into Hobart before you could see land. You could smell the plants. That's a really important psychological component for humanity, to be able to experience that environment, but potentially also has positive immunostimulatory components based upon microbial elements embedded in that system. Oh, and there's some more dogs in space, which I thought was cool. We've done a lot of work with trying to understand this and using mice as replicants. I'm really interested to see if we could explore animal experiments in space and getting animals and rodents. I know we have them up on the ISS. Very keen to explore the immunostimulatory effects of different component structures. We've done this for asthma. And I'd also be interested in exploring a smart toilet scenario. I have a commercial interest in this. We have a company we started, but we have built an opportunity to do continuous sampling of individuals in space. It would be very cool if we could apply that to every stool sample that was collected on the ISS by putting in place an automated sampling device that generated the data on site. Therefore, you wouldn't have to bring back feces in bags on a regular basis back from the ISS. I know we do that anyway, but it would be cool to have the system in place and allow you to maybe track what's going on in real time and provide real-time advice to the astronauts. It's a little bit science fiction right now, but we are exploring the opportunity to develop that. Thank you.